so Dr. Chari, can you give me an overview of the phase 1 study you are presenting at ASH and highlight some of its findings?
Sure, I'd be happy to. We've made a lot of progress in myeloma, but there remains the unmet need of patients who've exhausted particularly proteasome inhibitors, IMiDs, and CD38 antibodies, so-called triple class refractory. And so there's really a search for ongoing new agents in this space. And talquetamab is an agent that I think meets that criteria. It's a completely novel compound. It's the first time we're targeting GPRC5D, which is an orphan receptor. We don't know the signaling, but what we do know about is it's overexpressed in plasma cells. And pretty much other than that, some hair follicles, but very little of the normal body or other cells express this, making it an ideal target for monoclonal antibody. And in this bispecific antibody that we did this first in human study, while the expectation of a phase 1 study is primarily safety and dose finding, we saw very encouraging efficacy results for this population and a tolerable profile. So I think that's why this is a really exciting presentation, and I think it'll be great for patients and providers.
And why is GPRC5D a target of interest for multiple myeloma?
So there's already a lot of interest in this triple class refractory space, but BCMA is an exciting option. For example, there's targets of BCMA using antibody drug conjugates, bispecifics, CAR Ts. It's a very crowded space, but what happens when a patient exhausts BCMA-based therapy? And they also have their own issues with, for example, there's an infection signal. So there's a need for other targets, and GPRC5D meets that, I think, because it's a different target, overexpressed on plasma cells, not expressed on other tissues, and now we're seeing efficacy results on the same order of the bispecifics targeting BCMA. We're seeing 70%, albeit these are small numbers and early data, but 70% response rate in this population, and also durable responses. And so I think this is really exciting.
And what should hematologists and oncologists know about talquetamab as a potential treatment for multiple myeloma?
The drug is really exciting. We're seeing responses, and the safety profile, which is also of importance to practicing physicians and clinicians. Primarily from a hematologic perspective, we saw some neutropenia, although the neutrophil count for inclusion was only 1,000 for eligibility. Very few infections. In fact, grade 3 and higher infections, only 8% across the entire study. None so far at the recommended phase 2 dose. So that's really encouraging. CRS were low grade events. And we saw some dysgeusia and grade 1 and 2 skin toxicities. But overall, a tolerable drug with activity in triple class and pentarefractory patient population that's durable. So this is a really exciting option for patients and their caregivers in the future.
And what other studies will be looking at talquetamab in either phase 2 or in combination, or anything that's going on in the pipeline?
Well, the efficacy data needs to be expanded. So the phase 2 study is ongoing. There's also combinatorial approaches. We know that from preclinical data, bispecifics combine well with daratumumab, which is a CD38 antibody, but when we treat patients with CD38, there is a clonal expansion of T cells, which is thought to naturally synergize with these bispecific approaches. So basically a combinatorial approach with dexamethasone and daratumumab, and another one with the addition of pomalidomide. So triplet and quadruplet combination strategies are being studied. And obviously the other question is, once we have more data, how much earlier can we move this based on its safety profile and really encouraging efficacy? I think there's great interest in seeing this move up early. There's a lot of unmet needs in early diagnosis. For example, question would be high-risk myeloma, extramedullary disease, renal failure, frail elderly, a lot of populations where they don't necessarily make it to the umpteenth line of therapy. And so we need potent drugs earlier in the disease course.